So our second talk today is from Dr. Harmit Malik. I'll briefly introduce him to you. Harmit received his PhD in biology from the University of Rochester, where he worked on the non-LTR retroposons that can site-specifically insert the ribosomal DNA of most arthropods, and then those can be traced back to the early eukaryotic evolution. And as a postdoc in Fred Hutchinson Center, Cancer Research Center in Seattle, he focused on the centromere paradox, the extremely high rate of evolution at the centromeric DNA, despite the high rate of functional conservation. So he started his own lab at Fred Hutchinson Cancer Research Center in 2003, and as well as the centromere paradox, he focuses on the evolutionary history of host-pathogen interactions between the primate genomes and retroviruses and polyviruses. In his talk today, he will enlighten us about the concept of genetic conflict from the perspective of host-pathogen interactions. We're very much looking forward to your talk.
Thanks very much. Thanks for the organizers for picking my name out of a hat. I have to tell you, I've been talking to a lot of the students, and there's a fair degree of admiration for a lot of the speakers that are here. I just want to tell you that never goes away. So it's actually really a privilege for me to share the stage with people whose work I've basically followed since I started college and really have influenced my own thinking. So thanks for that opportunity. So I'm an evolutionary biologist, and we've not heard a lot of evolutionary biology in this meeting, but I kind of want to convince you today that you can get a lot of really cool molecular insights from evolutionary biology, and they can not only pose interesting questions for people studying cellular processes, but potentially provide answers that could be tested in a molecular genetic setting. So the concept of genetic conflicts actually comes to us from the fictional character, the Red Queen, who was introduced to us by the famous evolutionary biologist Lewis Carroll. And she says to Alice that it takes all the running you can do to keep in the same place, just because Alice didn't seem to be going anywhere after walking for hours. This was actually recognized as a very powerful evolutionary theorem by Lee Van Valen, who argued that in systems where two entities are actually competing for the same resources, where the fitness gain of one entity comes at the expense of the fitness loss of the other entity, you're basically always going to have this tug of war or this evolutionary arms race, which is played out by both parties evolving constantly just to maintain the evolutionary dominance over the other. And there's really no equilibrium to be gained here. There's always just this constant speed of innovation. You can imagine that kind of innovation happening in a host-virus interaction. So this is my high resolution crystal structure of a host-virus interaction which is happening between a host protein and a viral protein. And on the left-hand side of this cartoon, you've basically got a host protein that's now successfully recognized a viral protein, and because it does so, it can now cleanse this virus from the population. So the host is going to win this particular battle. This will put evolutionary pressure on the viral population to evolve away from this interaction. And what's remarkable is this evolution can happen in a very discrete step of even a single amino acid, exactly at this protein-protein interaction interface. Now you have a situation where the virus is winning, putting evolutionary pressure back on the host protein to innovate and essentially reestablish its antagonism of the viral protein. The net result is that this system is going to bounce back and forth between these two states with really no end until one of these parties goes extinct. And because one party is always losing this high-stakes evolutionary arms race, there's always going to be an evolutionary advantage to be gained by innovation. I'm going to touch upon two types of innovation today, but my lab primarily focuses on innovation in protein coding genes. And this is just a brief primer for those of you who don't think about molecular evolution on a day-to-day basis. We have a huge advantage in protein coding genes in that because of the degeneracy of the genetic code, nucleotide substitutions in a protein coding gene can either result in no amino acid changes, so these we call silent or synonymous changes, versus those that will alter the amino acid, so we call these non-synonymous or replacement changes. And so what we do is we make a simplifying assumption, which we know is kind of false, which is that natural selection views all of these changes as essentially neutral or cannot really see them because there's no net change being made at the protein level. Now we know that's not true, especially because the rates of translation and rates of protein folding can be affected. But compared to the selective cost or advantage associated with an amino acid replacement, this is still a very subtle change. And so what's nice is that in the same protein coding gene, I have two readouts. I have both a readout of the evolutionary time that has occurred since divergence, which basically roughly accumulates synonymous changes in a clock-like fashion, and I have a readout of how does selection view non-synonymous changes in this protein. If non-synonymous changes are in general deleterious, these will be removed from the population. If they are actually advantageous, they will actually fix in the population at an even faster frequency than these silent mutations. So what's nice about this is we can take a 10,000-foot view of genomes, and so here are some of our favorite genomes. So this is a three-way comparison of the chimp, rhesus, and human genome. We should probably pick a better representative there. So what we have is the total number of genes in the genome and this ratio of replacement to synonymous changes. And what you can see is that if, in fact, mutation was the only criterion, all the genes would basically line up with a DNDS of 1. The fact that they do not do so is because selection has profoundly affected this distribution. Indeed, for most genes in the human genome, purifying selection has acted on these genes, which basically means they have a dearth of non-synonymous or replacement changes because most of these changes were deemed deleterious by natural selection and were removed from the population, hence the term purifying selection. So what's interesting to me in my lab is actually this blip of genes on this right-hand side. These are genes in which selection has actually accelerated the fixation of non-synonymous changes because they were presumably advantageous. And not surprisingly, immunity genes are heavily overrepresented there because innovation at the amino acid level in immunity genes is something can be directly and rapidly acted upon by natural selection. So I'm going to introduce a case study of one of these sort of host-virus interactions in my talk today. And this is basically a situation where a viral protein is antagonizing some host immune protein, forcing the host protein to evolve away from this particular version, in turn forcing the viral protein to evolve to restore its antagonism of the host protein. So all of the work I'm going to describe today was done by Nels Elde, who's a former postdoc in the lab. And when Nels joined the lab, we had already established that this kind of evolutionary dynamic can be highly successful to even identify the protein-protein interaction interfaces between host and virus proteins, even in the absence of protein structures, simply by looking at what parts of these proteins actually evolve rapidly. And so this became a very successful paradigm to actually predict these viral specificity surfaces. So Nels was, of course, not satisfied with that. He wanted to increase the challenge for himself. So he considered the example where the viral protein was itself a stolen version of the host protein. So now you have two challenges. Not only do you have to evolve away from this particular viral antagonist, you now have to evolve discrimination between a protein that is itself a host protein and a viral mimic of that protein, which is basically evolving to look exactly like the host protein. So the model system we studied was actually the protein kinase R system. You've been introduced to RIGI already in this symposium. This is another one of those very immediate innate defense systems that is very important to defend against viral infections. If this organism is under viral attack, interferon is turned on, which will make an inactive PKR monomer. But if this particular cell happens to be under attack, like RIGI, it will actually detect double-stranded RNA in the cytoplasm and basically activate itself via dimerization, phosphorylating the translation initiation factor EIF2 alpha. And this phosphate moiety essentially ends up basically blocking protein production. So the cell's immediate response in terms of detecting itself under viral attack is, I'm going to block protein production because I don't want to become a virus production factory. If this block is not alleviated within a matter of hours, this cell will actually kill itself rather than basically become a viral production factory. Because essentially every cell in your body can mount this response, this can be a very, very potent block against an incoming zoonotic transmission of a particular virus. Now, what's neat about this is that several viruses have had to confront this because this is a pretty important block in their life cycle. And so they've come up with different strategies. Some viruses block the dimerization of PKR. Some encode proteins that hide away the double-stranded RNA. Some are very clever. They've stolen the EIF2 alpha phosphatase. So now you have a kinetic arms race between the host protein trying to put phosphate moieties and this viral encoding protein that's taking these phosphate moieties out. Perhaps the most clever invention comes from viruses like picornaviruses and hepatitis C, which have actually allowed PKR to block protein production, but have now invented an EIF2 alpha independent way of initiating translation, essentially turning the host protein against itself. Today, I'm actually not going to tell you about those strategies, instead tell you about a protein called K3L, which is encoded by pox viruses, and K3L acts as a competitive and non-competitive inhibitor of the PKR-eIF2 alpha interaction. The reason I'm focused on K3L is because K3L used to be eIF2 alpha. At some point in pox virus evolution, pox viruses basically stole eIF2 alpha and have basically turned it against the PKR-eIF2 alpha interaction by stealing this and now making it a mimic to block the very interaction that it was born from. So this can be